You know, it's just terrific to be here. It's really, really a pleasure and an honor. And the last time I looked at this building was a hole in the ground, so it's amazing to see this incredible structure. And the structure is populated by looks like a great group here, so it's real nice to be able to tell you something about what our Engineering Research Center is doing. We actually have our Engineering Research Center, NSF Center, is UWeb. I'm sure many of you know. We actually have quite a bit of activity these days in tissue engineering also, and so I could have talked about that, but I thought it might be more interesting for you to hear what is going on at the University of Washington in biomaterials, and I'll try and make this case about I think the very close, maybe intimate relationship between biomaterials and tissue engineering. So let's see, I got the nano word in this title, and actually it's a pretty good way, if you're given a poster or a talk at a meeting or something and you want to double your audience, just put the nano word in. It doesn't matter if it's relevant or not, just put it in.
I think it has our audience.
Well, it made the audience smaller. Okay, so you know, as you travel around let's say, the sort of a circuit that many of us in biomaterials go on around the world and in bioengineering, and there's lots of talk. It's called a nano this and nano that, and I just wanted to introduce where these nano ideas came from, at least my thoughts on it, and how relevant are they to my central interest, which is biocompatibility, and maybe how we can explore to use these nano ideas. That's really where we're going in this talk. And let me give some interesting credit to where I think some of the earliest work in nano came from. And this woman, Agnes Pockels, she is a self-proclaimed housewife, lived in Germany, and in fact took care of some ailing relatives in her house, but was fascinated by science and did a bunch of interesting experiments in her kitchen with soap bubbles and soap films. And it was a careful observer. And Agnes Pockels actually wrote a letter describing her observations to Lord Rayleigh in England. The letter was in German. Rayleigh had it translated, read the letter, and published the letter intact in Nature, which is, you know, something you can look up to for experiments you might have done in your kitchen. In any event, what her work really did was describe nanoscale thin film structures, and she had, based upon just sort of observations, careful observations and measurements, done a pretty good job of understanding the dimensions, the nano dimensions of these sort of things. So, you know, this was a true nanotechnologist well before her time. If I wanted to put one person before her, interestingly, it would be Ben Franklin, but I didn't have time to get into Ben Franklin's contribution to this. In any event, if one continues on this progression of where nano came from, you can talk about J. Willard Gibbs, and Gibbs, in his exposition on thermodynamics, described what an interface or a surface is. And he described this exquisitely well. There's nothing more to be done than what J. Willard Gibbs described theoretically in his work, which was published in that journal we all read, which is the Transactions of the Connecticut Academy of Sciences. Actually, it took a while for Gibbs to be recognized because he published in a not the best place, but although Connecticut, I think they like it. So anyway, Gibbs described sort of a surface and an interface, and again, the dimensions were nano. And then we had people going into the beginning of the 20th century. Zsigmondy in Hungary described colloids, and Irving Langmuir in New York described adsorption, lipid films, working with Catherine Blodgett, the Langmuir-Blodgett type films. So these were all nano sort of inventions that were going on a long time ago. But kind of, where did this modern nano thing come from? Well, obviously, people attribute a lot of this to Richard Feynman's seminal talk at Caltech in 1959. And Feynman said, you know, the principles of physics, as far as I can see, do not speak against the possibility of maneuvering things atom by atom. It's not an attempt to violate any laws. Feynman stimulated our ideas from work that really was going on. Nano engineers, as I said, were doing it well back maybe to the time of Ben Franklin. But in any event, Feynman opened their eyes to the possibility. And then someplace between 1959 and the year 2000, Bill Clinton, also at Caltech, gave a talk which was an incredible talk for a president of the United States. Bill Clinton, it was a visionary talk. Bill Clinton started to imagine the possibilities of compressing the entire Library of Congress into a volume the size of a sugar cube. Imagine the possibilities of nano machines treating blood and things like that. Just to hear these words from a U.S. president was really quite remarkable. But what went on between 59 and the year 2000? Well, it wasn't that the nano community was asleep. Between 59 and 2000, the molecular biologists and molecular bioengineers were quite busy. What molecular biologists was doing were discovering and even using remarkable nano machines. And I don't know if people have seen this amazing piece of work, but basically there's an ATPase molecular motor. This molecular motor has 6 protein subunits and a protein-like shaft in it. This is going on in every one of our bodies. The shaft churns, I believe it's 17 times a second, powered by ATP. It's an absolutely remarkable structure, and nobody, people have isolated it, studied it. Nobody really knows how it works. We don't have an idea yet, but it's an incredible nanomachine, and one group actually fabricated little nickel pillars on a nanoscale, connected them to the shaft, and made a little motor that spins around. This is an example of a real superb nanomachine, maybe something we'd like to do. And then sort of the science fiction writers, I don't know if people have seen this image, it just won a prize, but it's an absolutely remarkable image of maybe what we're daydreaming about in nano, some sort of remarkable gel-like nano machine that's going to come along and do some sort of interesting process. Although I'm sure most of us would say, why would we want to inject something in a red blood cell? We don't exactly know. But anyway, it made a great image. This is obviously an artist's conception of the future, and it says something interesting about nano. But we do have people like Ned Seaman who's taking DNA, and Ned Seaman doesn't really care about the fact that there are codes and signals encoded in DNA. He thinks it's a neat construction tool, and he uses the ability of G and C and A and T to connect to make incredible molecular and nanostructures. So just another example of the molecular and cell biologists, I think, doing remarkable things in nano before we used the nano word. Well, we have some sort of scaling relationships we're going to think about here. Again, a protein is a terrific nano machine. And in fact, Charles Tamford, who's one of the classic pioneers in protein biochemistry, and published very widely in the 60s, very influential work in the 1960s, just came out with a new book called Protein Machines or something like that. A very interesting philosophy on proteins as machines. But anyway, we have proteins, clearly a nanoscale structure. And then DNA. We have chains that are roughly 2 nanometers wide, but can be microns long. Interesting, the scaling in DNA. And we use DNA in devices. These are some linearized DNA chains that are actually done in our laboratory in a flow field. And then people are putting these into so-called MEMS devices, and people even, MEMS are microelectrical mechanical systems, but people are doing micro flow systems, and even thinking about nano flow systems. And these are, again, more likely at a nano scale or a micro scale, but we're, for example, putting coatings on these things that are indeed nanoscale dimension coatings that do things in these channels. And these things go into devices, DNA type chip. Again, now we're in the micron scale. And finally up to medical devices, which are clearly a micron scale, or excuse me, a centimeter scale in the case of devices like this. And how do these relate to nano? Well, as I see it, the ability to use nanotechnology to modify the surface of a medical implant device, maybe with proteins, is the key, I think, to the future. And we think it's clearly a nanotechnology. When we started our engineering research center, we didn't use the nano word. Little did I know we could have gotten even more funding if I'd used it. But anyway, I think we're doing nano, and how can we use these nano concepts in designing biomaterials? That's where I want to go. So let me give a quick introduction to modern biomaterials and ask how nanotechnology can help. And I always like to introduce modern biomaterials with this image. This is where I think the field of biomaterials, in its modern sense, started. And this looks like kind of a usual picture to launch into this idea. But this plane flew over the European theater in World War II, and you know, there were aviators, machine gunnists, assigned to sit here. And you know, where's the last place you really want to be sitting with the enemy coming down with machine guns firing? Is right out in front. As you might imagine, the injury rate was extremely high for these poor sods that got assigned to that spot. And after the war, a British surgeon was examining the eyes of aviators that got massive sprays of shards of plastic often in their eyes. And the eye is an interesting window into the body. So the aviator the surgeon just looked into the aviator's eyes and looked in there and saw these shards of plastic that were accidentally implanted years before into these eyes, and they were just sitting there. And he's looking in, and they're not inciting a reaction. The conventional wisdom at that time, this is a very striking observation, the conventional wisdom was anything like a splinter or a foreign body would incite a reaction. And here they were just sitting there. So he said, this plastic must be biocompatible. And he went to ICI Corporation and bought a sheet of this stuff and fabricated implant lenses for the eye with it. And this is what I think it was the first real observation of what we call biocompatibility today. And that observation, again, roughly 50 years ago, has led to an injury, led to an industry with quite a range of common medical implants. And I just have a few of them, you know, just kind of glibly illustrate your finger joint, or the infamous breast implant, or heart valve, or a hip joint, or artificial heart. Or here's this plastic lens that was made out of, in fact, to this day, many of them are still made of the exact same material that gunnery canopy was made out of, polymethyl methacrylate, the same kind of polymethyl methacrylate. So from a very interesting observation, industry has arisen, and in fact the industry has reached the point where it's entered sort of the public consciousness. And this is something I found in a newspaper personal ads, is mint condition male, 1932, high mileage, good condition, some hair, many new parts including hip, knee, cornea, valves, doesn't run but walks well. So you know, people just think very casually these days about all these replacements. It's not something that only scientists and physicians know. And another field, again, just to show these relationships that has sprung up is tissue engineering. Obviously, that's the major focus here, and a field of great scientific and technological excitement. I'd like to say biomaterials are important, even with tissue engineering on the horizon. And so I've shown this diagram to illustrate the connections. And you know, if you look at really the relationship, biomaterials now is a $100 billion market. Tissue engineering is still kind of near zero. Everybody says it's going to dominate the whole field. Obviously, a living tissue construct has many, many advantages over a synthetic biomaterial, but at the moment, it's kind of quiet from an economic standpoint. And biomaterials have helped millions. Well, there are a few people that now have skin products, but in tissue engineering of biomaterials are used. Both are looking to modern biology. Biomaterials has had steady growth over 40 years with really no sign of slowdown. And the tissue engineering, of course, is showing explosive growth and continues to do so. So there's interesting relationships between these two fields. But to get back to biomaterials, which again go into most tissue engineered constructs, we've had some success. We have worldwide millions of devices. I'm not going to read down the list, but there are millions of devices that go into humans. The U.S. healthcare market in 1998 exceeded $1 trillion, and it's actually pushing towards $2 trillion very rapidly if you look at the federal estimates of where the healthcare market is going. And we have millions of lives saved and the quality of life improved for millions more. Pretty impressive record. But another way to look at this is to ask how well biomaterials really do work. And again, these intraocular lenses, these eye implants, based upon that observation with the shards of plastic, yes, we're putting in 7 million per year worldwide, but there's a 25 to 50% reoperation rate on these devices, which is a huge cost to the healthcare system, trauma to the patient, obviously. The hip and knee prostheses were developed by a British surgeon in the 50s, and Sir John Charnley. The original Charnley hip implant lasted 10 to 15 years, and you look at the modern data, which come from, for example, a Sweden all-center study on hip and knee implants, and they report 10 to 15-year lifetimes too, on the average, through a whole country and through many, many years of data. There's still a lot of room, obviously, to improve these sort of things. Vascular grafts, blood vessel prostheses, were developed in the 60s, in fact, originally from parachute cloth, of all things. But the vascular grafts were developed as porous structures to heal, to infuse cellular regrowth and reconstruction. And to this day, we still don't get healing on vascular grafts. Heart valves calcify and thrombose. Percutaneous devices through the skin are supposed to seal to the skin. They just don't. Stimulatory electrodes encapsulate. Catheters have problems with thrombosis, infection, leading to thousands of deaths a year. Cardiovascular stents clot and close. Contact lenses lead to discomfort and even permanent eye injury. Dental implants loosening. The list goes on and on. There are, yes, successes with our biomaterials, but there are profound problems too. And so these are problems of much concern, but interestingly, where we have problems, we also have opportunities. Opportunities are suggested to do some new things. And we have to ask why the complication rate and what can we do about it. Sort of a central premise of the biomaterials field has been that the surface of a material dictates the biological reaction. Classic sort of model you draw is a biomaterial, and the surface adsorbs a layer of proteins. The adsorption is almost instantaneous, within seconds from biological fluid, and then cells come along and interrogate the material. So what do they see? They really don't see the biomaterial. They see some proteins that were driven by the surface. And so we have to ask, how does this surface that attracts the proteins dictate healing? And so we can sort of expand that diagram, and again, we have a biomaterial, and the biomaterial is implanted. That's really an injury. Implantation is an injury, and then the biomaterial goes into some body fluids and has a protein adsorption layer, and then some cells come along, like the neutrophil and macrophage, to interrogate the biomaterial. And these cells, in response to the biomaterial, are going to release some soluble components, cytokines, protein components typically, which go and talk to cells. And one of the things they seem to do is sort of talk back to the macrophage and induce the macrophage to form a giant cell. And this giant cell tries to encapsulate the implant or to consume the implant, to digest it, and really can't. And through a series of other molecules and cellular processes leads to the final result, which is a biomaterial that's seen in a capsule, in a capsular bag, a collagenous capsule. And this is a pretty dramatic scanning electron micrograph showing a Teflon device, and you can see this dense foreign body capsule and a much looser tissue outside. But this capsule is the interesting thing we get. These may even be macrophages at the interface, but it's a little hard to tell for an SEM. So anyway, to reduce it back to cartoons again, we have our implant, and you know, if you wanted a real definition of biocompatibility, you can look to the FDA, you can look to academics generating definitions. Ask a surgeon what he or she would think is a biocompatible biomaterial. And if this green thing, the biomaterial, is found within 1 month, is found within a thin, relatively acellular collagenous sac, and the reaction site is quiescent, the surgeon would say, this is biocompatible. And the interesting thing is it doesn't matter if this biocompatible biomaterial is titanium, polyethylene terephthalate, or polyurethane, or silicone rubber, or polyethylene, or polylactic acid. All of them heal in exactly the same reaction. So we had this model. The surface dictates the biological reaction. Yet the surface of these things is all essentially, the surface of each of these is quite different, yet the healing reaction is essentially identical in every case. Why is this? How could this be? Incidentally, we were wondering about the veracity of this idea. It seemed very radical in biomaterials to say that the surface didn't matter. So we decided to test the concept ourselves within our engineering research center. And we had what we call the UWeb implant group. But this is one part of that experiment. There was a hypothesis that said that the amount of the protein fibrinogen that absorbs to the implant dictated the foreign body reaction. The more fibrinogen, the more violent or more extreme the foreign body reaction or the inflammatory reaction. Actually, it was a very reasonable hypothesis. There was good reasons to think it was a good idea. So we tested it out. Each of these bars is a different biomaterial. I don't want to go into details about what they all are. Some of them will be featured later in this talk. But each of them is a different biomaterial, and this is the amount of fibrinogen absorbed. And one of the materials, it's a titanium treated by a process called the Kukubo process that actually makes it bond to bone. It absorbed a tremendous amount, 700 nanograms per square centimeter of fibrinogen. And then the other ones are on just a more compressed scale, but they range from something around 80 down to 10. So we have 700 down to 10 nanograms per square centimeter fibrinogen, a huge range of fibrinogen adsorptions to these materials. And we would predict this would correlate with a different foreign body reaction or healing. In fact, when we implanted them all, certainly within the error, they were all the same. There's one material that can be statistically different from some of the others. This is an osteopontin immobilized material. I'll talk about this one a little bit later. But basically, they all healed. This is the foreign body capsule thickness. They all healed just about the same, and in spite of huge differences in the surface properties. So what do you conclude when you get to this point? We have to sort of ask the question, we know that these materials are all different, but what is the similarity between all these widely different materials that make them heal the same? Well, our hypothesis is that they all have what we call uncontrolled interfacial proteins. The proteins that I talked about just glom down on the surface. They come down almost instantaneously. There's no control. We, as the engineers, have no control over this process. You stick it in and proteins glom down. Each material will have different proteins, but it's a mixture of proteins. The proteins are up, down, denatured, and they're out of our control. And that's the commonality. Yes, each of these materials does have different proteins at the surface, but the protein is a mixture, sort of a mess, something that nature never uses. Can we conclude that the surface properties do not matter? Well, if you looked at that set, you'd say, yeah, surface properties don't matter. But I think it's not a good conclusion. For one thing, biology does all its work at surfaces and interfaces. You may think that in fact processes happen in the bloodstream in solution, but it's essentially not true. Practically every process in the body happens on the surface of a cell or on the surface of an extracellular matrix protein. It's where I think 90% of the reactions going on in living organisms occur, not in free solution. They're surface and interfacial properties. Somehow biology makes a difference. Biology makes a difference with its surfaces. It's just our synthetics that are not doing it. And also, all in vitro biomaterials, all the biosensors and diagnostics do directly exploit surfaces. And as long as we're in vitro, we do see big differences in surfaces. So surfaces are important, but in different ways from what we originally thought. So the UWeb program, our Engineering Research Center, has a goal. At the moment, biomaterials heal in a capsule. In the future, we'd like to see biomaterials heal in a normal reconstructed, revascularized site, a reconstruction of the anatomy. Interestingly, normal wounds, you get a wound within your body, this wound heals just this way. There's excellent ability to reconstruct anatomy in wounds, but our biocompatible biomaterials are incredibly effective at turning off normal wound healing, and we're asking the question, why? So how can we get around this foreign body reaction? We think it's triggered by the nonspecific proteins, maybe. We think the macrophage cell, this interrogating cell, is involved. Normal wound healing follows a different course, namely the inflammation in normal wound healing is resolved on healing. Inflammation for an implanted FDA-approved biocompatible biomaterial, inflammation is never resolved. It's there forever as long as the implant's in. There's always some evidence of active cells and inflammation. So there's an important difference right there. The central UWeb strategy has really three components to it. One of them says that if proteins are the problem, maybe we can use stealth materials, non-fouling, sort of invisible materials that resist the pickup of protein. Maybe we can use these. At the other extreme, what we really want to do is encourage the specific interactions that turn on normal healing. There are processes that turn on normal wound healing, and can we, as engineers, learn about those and exploit and use those processes to make what we call engineered surfaces. But in between these two extremes, what we want to do is prevent the nonspecific interactions, always. Why? We've been doing it for 50 years in biomaterials. We know exactly what they give us. They kind of work for clinical implants, but we have to go beyond this. I think it's time to just get beyond this, especially thinking as an engineer. So we have our idea of an engineered biomaterial with the biology well understood. We're going to use the correct receptor interaction, and the bioengineers are sort of in charge of this reaction rather than some stochastic process. We're going to use molecules in defined orientation, conformationally stabilized, and maybe we'll need some bland, non-interactive regions in part of our design. It might be one of our design tools. To get there, there's a set of analytical tools that we use a lot. We're dealing with surfaces. The surfaces are on a nanoscale. The surfaces have nanograms or picograms of material that comprise them. We need some very sensitive methods to measure these surfaces. So there's a set of tools. You'll hear me mention them. I don't have time in this lecture to go into them. Things like ESCA, SIMS, atomic force microscope, contact angles. These are kind of our tool chest of methods to deal with these surfaces. Let me show you some data with one of the surfaces we worked a lot with. It's an attempt to make a protein-resistant or non-fouling surface. And we call it a stealth surface. And we use a molecule called polyethylene glycol that many of you are probably familiar with. People pegylate proteins to make the proteins invisible to the body. This is the chemical structure, very simple molecular structure. And PEO or PEG surfaces are found to resist protein and cell pickup. That's the observation. So we've decided we're trying to find a way we can make these surfaces that can be readily put on real-world medical devices. And our monomers to do this are some materials called glimes. This is a structure. It has this OCH2CH2 motif in it repeated 4 times and methoxy ethers on the end. Basically, it's a commercial solvent. If you need a tank car load of it, you can get this stuff. It's cheap and easy and reasonably safe. And what we do with this is we volatilize it and we put it into a glow discharge. This is not all that different from what goes on in a fluorescent tube, but it's a pretty energetic, ionized environment. We volatilize our glime, put it in here, and then we put a biomaterial in there. And we deposit a 20 nanometer coating on our biomaterial of this, let's say, energetically reconstructed glime that goes into the vapor phase here. Well, let's see what these coatings do. We talk a lot about the characterization. We spent years learning about the chemistry. I'm not going to discuss it other than to say they look very polyethylene glycol-like. In this experiment, our sort of control material is Teflon. What we're doing is just taking a piece of Teflon and coating it with this glime layer. And Teflon placed into fibrinogen, and this is fibrinogen adsorbed from plasma. So we have the plasma of blood and the plasma of physics combined here, two words. Interestingly, both coined from the same scientist, Purkinje, who also described conductive fibers in the heart. So there's interesting history there too. In any event, plasmas, the Teflon placed in plasma absorbs 116 nanograms per square centimeter fibrinogen, and then we treat this Teflon with our glimes. And we have some of them that have zero fibrinogen pickup. Now, we just can't do any better than that. This is pretty good non-fouling ability. This is measured with radio-labeled fibrinogen, which is a very sensitive method. We can't measure any pickup. That's good. We really learned how non-fouling these are using a technology called surface plasmon resonance. And what this methodology does is really measure a change in refractive index at a surface with great accuracy. And so if our surface is bare gold and we start adding some buffer, which is a function of time, we're going to add some buffer. And as soon as we add some bovine serum albumin, we see a huge increase, huge change in the surface refractive index. It plateaus as we reach a monolayer of albumin on the surface. And then we add a buffer stream again and try and wash it off. And we can't. It's irreversibly adsorbed. We take this gold, put it into our plasma reactant, put on a coating of glime. Now we have our buffer. We add the BSA, small signal. We add buffer, it goes right back down to baseline. This is called triglime, 3 of those ethylene glycol units. Tetraglime, that's the yellow bar, even less pickup when we add the protein. As soon as we add the buffer, right back down to baseline. These are pretty non-fouling materials. We looked at the adhesion of Pseudomonas aeruginosa bacteria, a sticky biofilm-forming bacteria. It's a nuisance in surgical problems. And a lot of data on this. It's from a pretty complicated experiment. But if you look at the tetraglime, extremely low pickup of these colony-forming units of this Pseudomonas aeruginosa. We've also learned to take this coating and use lithographic methods to photo pattern it. I'm not going to go through the steps, but it's a pretty conventional photolithographic process to make patterns. And in some recent work with Carl Boehringer, who's a professor in our electrical engineering department, and two postdocs in our group, we patterned this glime. And you can't really see anything. Again, the film's at 20 nanometers thick, so there's nothing to see on the surface. But what we've done is indicated with these dotted lines where we patterned it. And everything inside the dotted lines, we have removed the glime, so it's bare glass. And then we take this particular bacteria and we just seed the surface and let the bacteria sit there for 20 hours and then just be batted around by Brownian motion. And 70 hours later, where do we see the bacteria? It's on the glass where we've removed the glime. In fact, there's a movie here of this. Let's see if this works. Yeah, looks good. So these are images taken at every 2 hours. And again, we're just watching Brownian motion move these bacteria around. And you note where they're starting to wind up. I'm not going to go through the whole movie. There's a lot of frames in here, but eventually they all wind up on the bare glass and not on the glime. So this looked like the sort of material we really wanted, so we started looking at monocyte adhesion. The monocyte is the precursor to the macrophage. And sure enough, where we developed glime surfaces that had extremely low fibrinogen adhesion, they also had very low monocyte density. So this looked like a pretty good material. It inhibited these very sticky monocytes from sticking. It inhibits protein. It inhibited all the cells we tried. This looks like a great material. So one of our Ph.D. students, Ming-Xiao Shen, implanted these. And we got actually quite a surprise, which is opening up some new horizons for us. But this is an FEP implant. FEP is fluorinated ethylene propylene, a Teflon implant, the control. And here's the tetraglime implant. And this blue is the stained collagen. And in fact, if we look at the thickness of the collagen capsule for the Teflon and the FEP, excuse me, for the FEV Teflon and the tetraglime, they're essentially identical. So the ability to resist protein pickup our conclusion is we still think these non-fouling surfaces are going to be very useful in biomaterials of the future. They may be necessary, but they're not sufficient to give the healing that we need. So we need some active turn-on for the healing we'd like to see. And this leads into the next area we're working in, which is sort of the clues to healing. And we have a bunch of things that have shown potential to heal. There's a few things that have been seen in the literature. For example, hydroxyapatite heals very nicely into bone. It's the sort of healing we'd ultimately like to see. Some interesting work with porous structures and fibers. I'll show some of this. But in the blue area here, there are interesting molecules that are always present in healing wounds. And as soon as the wounds are healed, these molecules are gone. They're only present during wound healing. And this set of molecules is really something we're focusing quite a bit of our energy on at the present time. So I talked about porosity, and it's interesting. These are two identical materials. Here's one material, here's another, implanted for a month in a mouse. This is called a mixed ester of cellulose with very small pores or with much larger pores, but the chemistry is identical. And if one looks at the small pore material, there's a dense fibrous body capsule. There are active macrophages at the interface between the capsule and the implant. Classic foreign body response. Just change the pore size in this material, we get a very, very different healing reaction, a very open reaction. There are a number of blood vessels going through this capsule. So how did we achieve this very different healing reaction? Just change the porosity. That was pretty easy. That's an interesting clue, which we're working on. So one of my Ph.D. students is making a whole new class of materials that have very controlled pore sizes. These are very small pore materials at the moment, but he's making larger pore materials too. But he does this by templating the materials with microspheres and then extracting out the microspheres to get these very interesting open structures. Another interesting experiment on healing that's coming out of our Engineering Research Center, Professor Joan Sanders and her group, is electrospinning fibers. And she's noticed that as the fiber diameter goes down to below 5 microns, this is the foreign body capsule thickness after a month of implantation, and the foreign body goes down to 0. The foreign body reaction goes down to 0 when the fiber gets below 5 microns. So, in fact, you might even say nanoscale fibers seem to be almost invisible to the body. And Joan is now making mats and materials of these fibers for implantation. Is there a question?
Are those Teflon fibers?
These are polypropylene in this case. I think Joan would think that it's independent of the material. It's a size effect, which is what's seen with the pore size effect too. So geometry and pore size does seem to have some very profound effects on healing. The other class I mentioned are these interesting proteins that are present in wounds. They're called matricellular proteins. A lot of people talk about extracellular matrix, and a lot of people talk about cells. Well, these are the proteins that exist between the extracellular matrix and the cells. Cells don't really interact with collagen. They interact with proteins on collagen. Collagen is a wonderful way to deliver proteins. And so these are very important. And they've been named by one of our colleagues in our ERC, Paul Bornstein. They're called matricellular proteins. They are present during healing wounds. This is this purple-brown color is a stain for osteopontin, one of these materials. And at 7 days, one sees considerable osteopontin in a healing piece of Teflon. At 28 days, it's essentially gone. The wound is healed. So they are present in healing. And in some recent experiments, what we've done is, and this is our PhD student Stephanie Martin, has immobilized the osteopontin to, the material in this case is polyhema, just sort of like a soft contact lens. And if the polyhema is implanted, and this is actually has a lysine immobilized on the surface of polyhema, just to be an immobilization control for the osteopontin. At 28 days, we see a dense foreign body capsule. The blue is the foreign body capsule. Here's the hema. There are some features in it, but these features really don't have a lot of definition. We're not sure exactly what they are, but they're not terribly interesting to histologists. But the osteopontin immobilized hema, the capsule is riddled with blood vessels, more vascularized than we've ever seen a foreign body capsule before. So it's interesting, the osteopontin seems to encourage the vascularity that we want, but also does not seem to inhibit the foreign body capsule. So the osteopontin as a protein seems to do half of what we want. But then we have another observation on another protein called thrombospondin. And this protein, what we've done in this experiment is to use a knockout mouse that has thrombospondin knocked out, and looked at the, we have two materials, a silicon rubber and an oxidized silicon rubber, and we look at the vascularity and we look at the capsule thickness, and it turns out that in the thrombospondin knockout mouse, first of all, we get 10 times more vascularity, and the capsule, the foreign body capsule, it actually looks like it got thicker in the thrombospondin knockout mouse. But if you look at the histology of this capsule, and I didn't include those slides in this talk. Again, it's a very open, diffuse capsule. Yes, we measured it as thicker, but there's very little collagen in it. It's a very, very different morphologically from this one. So it seems like the thrombospondin is sort of the enemy. Where thrombospondin is present in the wild-type mouse, we get this dense foreign body capsule. Where the thrombospondin is absent, we get a much more vascularity and a much looser capsule. So what we're trying to do now, in fact, one of the goals, one of the engineering goals in our engineering research center, is to develop a sort of a systems approach to using osteopontin, which increases vascularity, and the thrombospondin, which reduces the capsule. How can we use these both to generate the sorts of signals we need to turn on a normal vascularized healing rather than a capsular healing? And badly running out of time here, so I'm going to zip through a bunch of slides and just kind of tell you generally what I'm doing. I won't have time to go into the details, but one of the ways we're talking about delivering these protein signals that we need is what's called templating. What we do is go through a series of steps where we literally make a material with pits. We use the original protein as a template and have pits in the material. And these pits have recognizability for the proteins. And interestingly, this purple color or this pink color is sugar. So the surface of the implant becomes a sugar surface that has a recognition sort of stamped into it, templated into it, that allows the sugar surface to recognize proteins. The idea is that the template protein aligns the sugar or hydrogen bonding array on the sugar, which is cross-linked in place in this binding pocket. And we have some evidence that the process we use has the fidelity to make pits the size of proteins. These are glutamine synthetase pits examined with a transmission electron microscope. You can see from the histogram that our pits are just about the same size as the protein. So we have the fidelity to make these nano pits. And we can use AFM again to sort of look at what our surfaces look like. Obviously, rougher fibrinogen has coarser pits, and if you want to use your imagination, you might even think you could see some fibrinogen there, but I won't push that point. In any event, what we get with these pitted surfaces is a remarkable degree of recognition. In this experiment, we're comparing lysozyme and ribonuclease, both 14 kilodalton proteins, the same size protein, and basically ribonuclease pits recognize ribonuclease about 20 to 1 over lysozyme, where lysozyme pits recognize lysozyme over ribonuclease about 30 to 1. So these surfaces have remarkable selectivity, and they're just sugar. And this work was published in Nature in, I think, 1999, and we have some students continuing this process with things like fibronectin now. To show this recognition, in fact, we used a process called microcontact printing. We made a rubber stamp at a micron scale, and the ink for our rubber stamp is protein. And in this experiment, we inked our stamp with streptavidin protein, stamped it on mica to make this sort of pattern of streptavidin, that's the green, backfilled it with bovine serum albumin. So now we have bovine serum albumin islands in streptavidin. We used this as the template to make an imprint, a sugar imprint, and then we dipped it in a streptavidin bovine serum albumin solution. And this is an AFM image of biotin to which was attached to 10 nanometer gold microspheres. And the biotin goes to streptavidin, and you can see where it goes. It goes in the spaces between these holes in the stamp. So the surface is all sugar, yet one part of the surface likes albumin, one part likes streptavidin. This might suggest a method to make a surface on a biomaterial that doesn't involve immobilizing an expensive, fragile protein. Instead, we could just use sugar. But the problem is our proteins are every which way. They're up, they're down, they're sideways. Those proteins, we put on mica to make these imprints. What we really desire is something like this, so we can make a really good imprint that would bind the protein with the, for example, the interesting binding or reactive site or ligand binding site was facing out, so we'd have real control of the proteins at the surface. And we're using the SIMS methodology, surface analysis methodology, to study the orientation of proteins. I'm going to skip through a group of slides here now and go just to the conclusion of this story. But basically, we're using protein peaks and SIMS to get to where we want to go, and using some multivariate statistics. And let's see, you might just go right to the end of the story here. Basically, we can distinguish using the SIMS if the protein is up, if the protein is down, or the protein is randomly absorbed by this clustering of different multivariate correlates of the points. And we think that if we had the proteins, we know now that we can align antibodies all in such an orientation. And if we can do this, if we can use this alignment in this way, which we found we can get a monoclonal to a different region on a protein, have the protein stick in an oriented fashion on this IgG, and then template that. So that's one of the places we're going. We're also mobilizing type 1 collagen on surfaces, which does bind the proteins in a very specific way. And so instead of having random proteins on our biomaterials might have the type 1 collagen, which then binds the osteopontin in a very controlled way. And these experiments are ongoing now. This idea of protein delivery, just look at what a virus does with its coat proteins. It delivers the proteins with organization and specificity. We're just trying to copy the way nature does its protein delivery, the precision with which it does. And we have a vision. Maybe someday we can take, just shown with the hip joint here, coat it with nanopits, implant it in bone. The device concentrates the proteins that we, as the bioengineer, want to bring to the surface and leads to improved healing. This is a sugar-coated medical device, and the patient's own proteins are used for healing. So these are some of the ideas that lead to what we're calling an engineered biomaterial, an organized, ordered biomaterial, sort of describe that. And I see an interesting progression. Of course, we've used biomaterials for a long time, and I think we're going to have biomaterials in the classic sense, the silicone rubbers and the titaniums and all, at least for another 20 years. I'm sure we'll be seeing them widely used in medicine. But maybe by 2010 or so, maybe these precision nanosurfaces will start appearing in medicine and controlling the healing response. I think tissue engineering is certainly the wave of the future. And then beyond that is going to come true regenerative medicine that's going to, for obvious reasons, replace all that. So there's a quote from Voltaire, look at this, is early 18th century. The art of medicine consists in amusing the patient while nature cures the disease. I think the intrinsic healing mechanisms in our body are really what's important, and the physician's job is just keeping the patient happy while the body does the healing. I think a real future, this is a cartoon obviously, but maybe some of you read about this experiment of putting electrodes in the brain of a monkey and having the monkey control the cursor on a computer with just electrodes implanted in its brain. This has incredible potential, for example, to allow a paraplegic to walk, just controlling with the person's brain, maybe some prosthetic device in the legs. The potential is incredible. We're doing things with this idea. What's the problem is the electrodes encapsulate at the moment over. If we could get healing electrodes, I think we can make devices like this that really work for long periods of time. So let me finish up. This is about half of our UWeb program. I couldn't just couldn't get everybody together at the same time for the photograph. But anyways, a lot of people that obviously have to be thanked for all this work that went into it. And so thanks to my students, staff, colleagues for this work. Thanks to Bob for the nice invitation to give a seminar here. And I'd be glad, please take some questions. Thank you.
Thanks, buddy. That was great. Let's open it up for questions.
Yeah.
Okay, I wanted to ask you about the data you showed from the osteopontin fixed to the material surface.
Yeah, yeah.
In that case, is the osteopontin basically, is it just fixed to the surface where the cells just at the interface see the osteopontin, or does it diffuse out to where the cells sort of in that encapsulated layer all?
It's at the surface. It's a mobilization technology called CDI, carbonyl diimidazole, pretty classic immobilization technology. And as we describe it, it looked like it was pretty effective, but the molecules were we call it sloppily handled. They weren't controlled for orientation or conformationally stabilized. They're just glommed down there, yet they still worked. So we think we can do much better. But they weren't out on tethers, for example, or they weren't diffusible. That's what you mean.
Do you have a feeling then for how that actually stimulates angiogenesis in that case, as far as, do you think it's an attractant for endothelial cells and macrophages, or it's...
Yeah, those are very good questions. There's actually a lot of fundamental biological data on the processes stimulated by... but one of the things it does do is sort of calm inflammation. It seems to reduce the magnitude of the inflammatory response. And we think by reducing the classic, macrophage activation, we think by reducing that, it gives more opportunity for normal healing or reconstruction. So it may work. It may increase the angiogenesis by reducing the massive inflammation. Yeah. In making the porous materials? Yeah. It's a classic problem, and we sort of have some interesting approaches. One of them is to make these materials. You know how you might flour a breadboard before you put bread in to keep from sticking? We put our microspheres. We make it with microspheres. We put our microspheres down on a breadboard and put this dough of polymer down on it, so we have clear inroads into the material without the skin. It's tricks. You got to do tricks to get around that skin, but it's thermodynamically it wants a form, and it's a problem for porous materials. These are polymethyl methacrylate, and actually it was kind of hard to find a source of soluble. Most microspheres are cross-linked. We needed soluble ones that we could dissolve out, and we finally found some sources for them. Yeah.
Other questions? Gosh.
Do you have a sense for how stable some of your surfaces are in terms of chemical desorption, or perhaps in response to fluid shear or just chemical gradients? It seems like that's one of the critical problems, especially in extending the life of some of these implants.
Yeah. Well, the immobilized protein surfaces are as stable as the proteins, which is stable and unstable both. In shear, for example, if we're covalently immobilizing, these are stable against the shear forces, but maybe the shear can denature the proteins. We don't know that for sure. The sugar surfaces, they found sugar in the tombs of the pharaohs, intact sugar. Sugar is very stable, actually, if nothing eats it. And so we're hopeful that we can make implants that could be stored for long periods of time with sugar on the surface.
There are some of these materials that will never get really integrated in the body. So what's your vision about what's the perfect, let's say, reaction to a material like that? Would you look to a specific thickness of the capsule, a specific composition? I mean, obviously you don't want inflammation, but what else are you looking for?
Well, I'm not sure exactly. To take an example of a material that maybe you'd say would never, or a device that would never get integrated, let's take a pacemaker, okay? The pacemakers are given considerable problems by this thick capsule. The surgeon has to dig through them when they have to replace or work on the pacemaker. The leads get heavy fibrous encapsulation. If we could reduce this, just even reduce it, I think we'd already make a contribution to surgery and medicine. Also, a lot of device-centered infection is associated with the lack of vascularity. With this vascularity, the body has mechanisms to deal with infection. If you cut off the vascularity, the body doesn't have a way to deal with infection. So we think if we can improve the vascularity around the site is where we're going. I don't know if we can ever totally eliminate the foreign body capsule, but we know we can reduce its thickness, its density, and improve its vascularity, and that's kind of where we're heading. And that would go for, I think, all medical devices. Implanted devices of synthetic materials could benefit from this technology. In fact, there's no reason to believe that the increased vascularity and the reduced inflammatory response wouldn't be good for a tissue engineering matrix, too. A tissue engineering scaffold should benefit from similar kind of protein control and technology.
Is there a final question? Anyone want to? I think, buddy, Steve?
How would you use your strategies, let's say, in a person that doesn't heal well, like, for example, the diabetic patient? How would your strategies be?
Now, it's a really good question. And, you know, if the answer is maybe the diabetic doesn't heal well because the diabetic doesn't have enough osteopontin. Again, these implants are like affinity columns for osteopontin. You know, in principle, the surgeon could dip it into some osteopontin and then implant it, and that might improve the healing. I think we'd have to know, and this is an ongoing question. I know we have people right in our Engineering Research Center working this to ask why diabetics don't heal or why they heal so poorly. We don't know exactly. But if we knew that, we might be able to develop biomaterial strategies around that.
Your comment about osteopontin was more a hypothesis, maybe, that...
Yeah. Well, I think there's no doubt that osteopontin is very important in the healing of normal wounds in normal individuals, and so we feel it could also help to heal biomaterials. But maybe a diabetic is short on osteopontin. We don't know for sure. You know, we don't know. For example, it's an extremely good question, and it's a question that is on the table. It's very hard to do the analysis. Once the body puts all its stuff on, we don't know what's happening underneath. Very hard to figure out what's happening inside or at the surface of an implant that's been implanted for a long period of time. So we don't know exactly. You know, there's two things we have, two ideas we have. One, that the proteins may stay there and stabilize things. Another, that once they've done their work, they may get digested and vanish. And if they've done their work, that's probably fine too. The proteins? Yeah, the body has great mechanisms for recycling proteins. Yeah, again, we don't really know for a fact how long those proteins on biomaterials stick around. It's just not known. And it's just analytically a very, very difficult problem to get at.
Ben?
I wanted to ask about the sugar coating of the abdomen.
Yeah, strep abdomen was the, yeah.
How does that turn out for more conformationally sensitive protein, like you mentioned,
Yeah, and actually we did it. We got some very, very impressive results with fibronectin. So our first thought was to use small, durable proteins like the lysozyme and ribonuclease. And those were good cases, and they worked out. But we've done it with fibrinogen, IgG, albumin. Now fibronectin is the latest set of data to come out. And, you know, it seems to work. There's no doubt that the more we damage the conformation of the proteins, the less active our surfaces are. So some of the techniques to stabilize and organize proteins at surfaces are going to lead to, we believe, to much better imprints.
What type of surface were you absorbing the fibronectin?
At the moment, all this work is based on absorbing to mica. And there's some evidence in the literature that mica is not terribly denaturing for proteins. And it's also molecularly smooth. That's the advantages of mica.
Julie, you want to ask the final question?
With your Lyme surfaces, what have you found, like you showed protein fibronectin absorption, what about for platelet adhesion or activation in particular, as well as lymphocyte?
Okay, well, good questions. In fact, one of our Ph.D. students, who's going to be just submitted an abstract to the Society for Biomaterials meeting this year, is coating the inside of tubes with these Lyme plasma coatings. And in fact, we're working with Mike Sefton and the Toronto group on assessing platelet reactivity with these materials. This is a very interesting area because Tom Horbett has this theory that, again, fibrinogen pickup is directly related to platelet activation. And these don't pick up any fibrinogen, we know that. But we know they have this interesting interaction with neutrophils I was telling you about earlier. So we're kind of fascinated to see how they're going to react with blood. And we should have some real data soon. It's a major technological accomplishment to be able to coat long tubes, and we can do it now.